I'll just leave it here. Yeah, I know it needs to be cleaned. Where? Yeah, okay. Oh, Mena of the upper? Oh, you mean to be, Mena be you, you mean? Oh, that's, okay. I'm not there. No, it's not. It's not. No, take it to your freedom. Take it. Okay. Yeah, that's wrong. Bye-bye.
Sounds good. Thank you.
Check my time. Hi, we're going to wait a couple of minutes. There's some people getting badges and coming along in, and then we're going to get going. So welcome. Thank you for joining us today. And we are very excited to have these presentations. today. You're in for a treat. So hang in there a couple of minutes and we'll get started. Thank you. said it.
Oh. Yeah, so you just, you can, if you run into any trouble, just hit ESC and then Control Command F.
Okay. Thank you.
No problem.
Okay.
Isabel, do you have your bell? Do you want me to grab it for you? Okay. Yeah, it should be the one with the registration table.
Okay, we have some people still coming, but we're going to get going. Welcome. This is the event we have been working toward for many, many months now, and we're thrilled that you're here. Internet access information and social media information is here. We are very, very pleased to be back here. This is the 11th time we've had a chance to run the Pitch Day event, and we're thrilled to be back at Florence Gould Hall. We were here last year and just love the group that's here. We love the environment, and you're going to love the reception, and especially the food. That's going to come at 4:30, so hang in there, and there'll be some things to eat. Deborah Novick is here. Nick Gunkel is on the stage right now. They are going to be the moderators for the pitches this afternoon. My name is Mary Howard. I'm the program director for the Westchester County Bioscience Accelerator, also known as WCBA, and the Accelerator for Biosciences in Connecticut, ABCT. So here's a quick agenda of what we're going to be doing. We're starting 12 minutes late, so we'll catch up on my intro, and then we have a treat. Joe Landolina is here to join me, and we're going to have a fireside chat about New York and how Metro New York has changed and how bioscience has changed and the role of founders in all of this change and what some of our challenges are coming up. So we're really looking forward to having a conversation with Joe, just a leader in so many areas of biosciences and business and a human being, and we're thrilled to have him return. We have two groups of pitches that are here. The first is going to start, and then we'll have a break. As you came down the escalator, you saw some easels, and in front of the easels, you'll be able to connect with the people who are in the first, what we're calling the first flight of pitches. So that's an easy way to connect with them after they pitch. And then we will be ringing some bells. Isabel will be here, and we did not give her that task because she has Bell in her name, but she is going to be ringing bells to get everyone back. And then we'll have the second flight of pitches, and then we'll have some closing remarks, and then we'll head on upstairs to the 8th floor to the reception. I just want to say that this is an event where people have been working. They were selected because they have high potential intellectual property, science breakthroughs. They've got commercial potential. There's an interest that external reviewers believe in their interest in what they're doing strategically to make a difference down the road. It takes a long time to get these ventures off the ground. So we just ask you as the audience to think about what you can do to participate in this. One of the fun things about being part of this community is helping make connections. Those connections aren't necessarily investors. They may be partners. They may be people who could join one of these companies. They may be organizations that could use some of the expertise of someone that you know. So please think about that and be thoughtful about approaching people, even if it's scientific area that you're not familiar with, because there's a lot of opportunity here, and we're going to share some of the history of where these people have gone on to. Just to give you a background, the programs that we had the good fortune to run, we were Design Technologies. We rebranded in January as First Time Founder, reflecting the customers that we have. Those ventures have gone on. We've had the fortune to work with a large number of them in the Metro New York area. There's about $2.6 billion, over 1,000 jobs have been created from these ventures, and we've relaunched most recently the ELab program as a series of master classes, and we're so thrilled to have the first two done. We'll be doing the second one, the second anniversary one of due diligence this summer, so you'll hear about that. but more, we've often been asked, okay, what happens to these ventures, right? Where are they now, right? And so we have a new way of showing that this year. We're showing this by cohort year. And we want to remind you that this cohort, if someone stopped in 2022, they've really only been out of the program for less than a year. And so in Westchester, we're seeing over 62 jobs created, 17 million raised, and just people already selling. Already non-dilutive funding, 4 SBIRs awarded. We know there are a couple more that are on the way with the scores that people have received, and then quite a number of people already receiving funding. So these are people who started in the pandemic, continue to grow their business in the lockdown, and are now the first group that actually pitched in person was last year, the 2022, and Maya, congratulations on the $4.2 million that you raised for your digital health platform for organizing surgery. We hope we don't need it, but we're grateful that you're there to help us get through it in a more safe way. In Connecticut, we have another 2 years of ventures who have come through. Again, extraordinary 245 jobs that have been created. Every time we update this, the jobs are going up. We had our first exit, which was ELabNYC, a therapeutic venture that was sold. But quite a number of people are receiving SBIRs. In fact, 20% of all Connecticut NSF SBIR awards are going to ABCT alums. So, you know, very interesting, very early stage, nationally validated funding that's going into these ventures, and a lot of additional funding coming in afterwards. So we wanted to talk about, you know, what have we been working on to try and support these people who increasingly are apparent that they are just really an extraordinary group of people. So we've had the chance to work with Solview, who is a member of the ELab 2013 cohort, a venture that has gone on to doing a lot of bioinformatics programming for drug discovery companies, and we asked them to take some of the data that we had and help us visualize it and work with it. So this is actually a representation of the mentor needs that are collected weekly. And it has allowed us, and that's actually what you're seeing on the top of these banners, you're seeing literally the one-to-one descriptions of what the coaches are sharing the needs are every week. And in a representation grouped by types of needs, scientific, business, clinical, et cetera. And as you can tell, there's a lot to this, and that has been something that we've spent a lot of time trying to support the mentors that we're so fortunate to work with, to be able to provide them some more specific information about what we hear from our coaches is needed. So the mentors have an extraordinary breadth. There are over 275 of them. We are very fortunate. The group that is highlighted here are mentors who've been working with us for many, many years. They come from, as you can see, many different areas of expertise. And there's quite a number of entrepreneurs as well, and the entrepreneurs have a lot of different backgrounds as well. Those mentors and the needs that we're trying to map to support them are supported importantly by a number of corporations. We've been very fortunate in the ABCT program to have a broad spectrum of expertise joining and supporting. We call them corporate supporters, not sponsors, because in addition to the funding which they provide, it's a lot of time in mentorship, in education, in workshops, in virtual office hours, and we're very, very grateful for them being here. And you'll also notice that a number of these organizations are also supporting the Westchester program as well, which is wonderful for us, and yet the communities are not the same. We have different organizations that are focused in different communities, and that's one of the things that's important to us. We try to create a balance of corporate supporters who bring their expertise to this We deliberately recruit people who are leaders in their field to help support these very early stage ventures. And we have been very fortunate to work with a number of them from the very beginning of these programs, and we're very happy to welcome back Moses Singer and Linda Malik, who was involved from before the beginning of the ELAB program, which started in 2012. So 11 years of having a chance to work with people who are committed to a long-term vision of building an ecosystem in New York. I want to thank all of the people who are been part of this. You should take a lot of pride in this impact and building these communities, because this is a massive effort that a lot of people are bringing a lot of goodwill towards. So thank you very much for that. I want to take the chance now to have a conversation with my friend Joe Landolina. So I'm going to ask him to come on out and share with you some perspectives of some of the changes in Metro New York and globally since 2012, when I first had the chance to work with him. So please join me in welcoming Joe to the stage. Thank you.
Thank you, Mary.
It's great to see you. I think maybe there's a little bit of feedback. I'll see if I can do something. But Joe, I've known you since 2012, and a lot has changed in Metro New York since 2012. But I think for those who haven't had the pleasure to know about you and your background, I think it would be helpful to share a little bit about where you started back then and what you're doing now, to give some background to that.
Sure, of course. So I'm a chemical engineer by training. I was lucky enough that when I was in undergrad, I discovered technology that stops bleeding rapidly. And we founded a company around that called Cressalon that was spun out of NYU back in 2010. And so over the last 13 years or so, we've grown Cressalon from what was effectively an idea in a dorm room. We were part of one of the first cohorts, or the first cohort for ELAB. In 2013, and after that we started growing really rapidly to a product line that is now in the animal health industry. We're being sold in more than 30 countries around the globe. We have imminent clearances coming up for human use. A team of about 75 engineers working here in New York, and today Cressalon is the only aseptic fill and finish operation, so meaning the only manufacturer of sterile medical devices at a CGMP level in the 5 boroughs of New York. We operate about 25,000 square feet of clean rooms where we do A to Z production for all of our global markets.
So, Joe, one of the things that's always been so extraordinary about hearing your story as it's evolved has been the number of areas that you continue to be a pioneer in. I remember the discussion with people about there being a GMP, CGMP manufacturing facility in Brooklyn, and people just couldn't even believe that that was in existence. They thought that I was talking about something that was somebody else's idea. But you know, you have seen an evolution in a number of other areas. With your involvement continued with NYU, you're a huge supporter of entrepreneurship at NYU. You've been a big spokesperson for biosciences with the city of New York, with the state of New York. And I know that you have been involved even to get that power in to do the GMP manufacturing facility with Con Ed and other organizations, because there isn't power to do some of this stuff. So, you know, tell us a little bit about where the community was back in 2012 and what you see has changed.
Of course, it's been a drastic change. I think when we first started this, the community was very much academically driven, and so we have a lot of great universities in New York that have great people and great ideas and great research, and the market in New York looked like there were corporate offices for large pharmaceutical companies, and a lot of great ideas and startups that were happening at the academic level in basic research, but we didn't have a full-fledged community. And there are a number of reasons for that we probably don't have time to go into in a lot of depth today. And one of those is that it's really hard in New York, and New York is expensive, and it's difficult to scale past the point of just being at the bench, and there weren't a lot of resources from an investor perspective, there weren't resources from a talent perspective, or from a government perspective if you had a great idea and you wanted to start growing. And with us, and what that translated into is that, I mean, our first manufacturing site, our first offices, we bought a building that we found on Craigslist. And just it wasn't like we went through a full selection process. We weren't partnering with any of the logos that we may see sitting around on banners at events like this, because it didn't really exist at that point, or at least in the form that was needed to scale. And what I love to see now is that over the last decade, we've really started to put those resources in place. And if you want to take an idea from A, maybe not to Z, but if you want to take an idea from A to X or something like that, those resources are available now, and there's a lot of work to be done. And one thing we were talking about before this is making sure that we're not just building a startup economy, but we're building a biotech community within New York that is 360 degrees, and that includes every employee of a full services life sciences industry, and not just supporting founders and engineers in doing lab research, but making something that's actually sustainable, where we can start to fund back to the communities that we're part of.
Well, I want to talk about that because in the cohort, when you're starting up, you're trying to live to fight another day. And I just want to get a sense from you, when did it become apparent to you that there was a broader, a need to think more broadly and to be involved more broadly, and how did you start getting involved in thinking about this community and what the community needed?
Sure. So, I mean, again, as an entrepreneur, your goal is to stay alive. Your goal is to do effectively the minimum or the least burdensome path to get to the next level of the business. And for better or for worse, the technology we were working on is not easy to play with. And because of that, that meant that we couldn't outsource, we couldn't partner off, and we were forced from a very early stage of the business to start thinking about having a very verticalized company. And in doing that, we kept getting called crazy, or we kept looking up for resources that we assumed should have existed, but then we realized did not exist. And I tend to be very opinionated and very vocal, and so we started putting ourselves in the rooms where those decisions were being made. And I mean, things like realizing that a modern clean room requires more power than is supplied to a standard commercial real estate campus is something that no one thought through in the very beginning, but no one was going to solve that for us either, and we had to find a way to get the right parties and to make ends meet, because at the end of the day, all of these decisions were not, we'd prefer to be in New York and we have a cheaper option somewhere else, but because we like New York, we're going to stay here. It was, we have to do this, otherwise the business will fail and we won't get our products onto market. And that's one of the most powerful drivers there is. But thankfully, we've been able to forge a number of those paths that now companies can and are, frankly, following behind Crestlon.
Well, I think that's really true. I mean, the idea that there wasn't enough power to come to a building in Industry City was, I think, a shock to a lot of people. And it wasn't anywhere near enough power. So it was a pretty major accomplishment for a very, very small company to get that done. When you think about the community, I'd like to just talk some more about that and where you think some challenges are now and where founders should be thinking about getting involved in the community, because these, we've just come through 3 years of a pandemic. There's been a lot of focus on health. There's, very different priorities right now. So how do you think about getting involved in the community? You talk about building something sustainably that's not just for the engineers, the scientists, the founders, but for making a sustainable venture. So how do you think about that?
Sure. I mean, it gets back to what I mean by a 360-degree community. And so what we're working quite a bit on today is that a life sciences company, that has true scale is not just an engineering team or a research team that's sitting in a lab. And that may be the major focus, because those are the expensive and the hard things to do. But at the end of the day, we have warehousing, we have trucking and shipping, we have supply chain, we have manufacturing, and those roles range from hourly employees all the way up to senior management. We have sales forces, and these are pieces of the community that today, if you look for preclinical research is a great example, right? Outside of the academic sphere, if you're looking for to do preclinical research, it doesn't really exist here in New York in a robust way as you would see in other markets like Boston or the Bay Area or San Diego. And so we have to ask ourselves, why is that? And how do we as stakeholders in this, especially those of us that are stakeholders that are further along in this process, how do we develop those talent pools, because these are lucrative jobs, and they run the gamut. And Crestlawn, we've made quite a bit of an investment into Sunset Park, which is the neighborhood that we came up in. And the question that we keep asking our teams is, how can we make sure that as we're growing the business, we're offering job opportunities to local Brooklynites? And it turns out that there are lots of people that live in Brooklyn or in any of the other boroughs of the city that end up commuting into Jersey or into Long Island, or further afoot to take jobs, because they don't realize that this type of community exists. And there may even be another layer, which is that there's not a lot of great education or workforce readiness programs in the city. And that's something that I've been somewhat vocal about, and that areas like North Carolina have done really great jobs of building workforce development programs that allow people to realize that, and most of my employees, as an example, start as being pre-med students, right? They want to be a doctor because they don't understand that there's an entire industry behind it as high school students. And then they get into their roles, and many people in this room may even fit into the same bucket, and they realize, you know what, there's something more interesting in this industry, or maybe I don't want to do exactly this, and then they realize, well, you can go into industry, and once you do that, then there's quality roles, you have manufacturing roles, you have production engineering and R&D and all the other flavors that come along with that. But we have to open that up so that students and young talent are able to be educated to come back into these types of industries. And one of the biggest challenges as well will be that in a post-COVID world, you can't make a medical product from remote work, and so a lot of these jobs need to be here and on site. And there's a challenge with that as we look to grow the industry and community here in New York. But there's also a benefit, and it means that individuals that may not have opportunities in certain industries can have highly paid, lucrative opportunities working in roles that don't require an advanced degree. And we haven't built that in New York just yet.
So what do you think about the kind of timing of when that might be coming about, or is that sort of TBD if that can happen? So
I think it's baby steps, right? And I mean, companies like Crestlawn hiring and training means that our employees, when they're no longer employees of Crestlawn for whatever reason that may be, are now hireable by other companies in the space. And if we go back 50 years, when Pfizer was founded in Brooklyn, they started in Flushing Avenue. A number of our employees were employees at the Flushing Avenue site that got displaced outside of the city but still lived here, and now were able to come back. And so it's about having enough companies in the space, about having that start building that and charging forward regardless of what programs may be there. And then the next step is working with local community colleges and working with the government to put programs in place that incentivize and that drive awareness that these types of roles exist.
Well, I think a lot of the ventures who are here today wish they were in the position you're in to have the kind of problems you have with hiring
Tell me
people
about it.
But it's coming sooner than you think. It doesn't seem like that long ago when we saw you and Isaac building out your your plans and hiring all of your first employees. And it's been very, very exciting to see where all you've come. So how many employees do you have right now?
So we have about 75 employees, and to the point of not having a larger community, we have about 50 open positions on that team. So if anybody here has any friends that are interested to come work at a fast-growing biotech company, look me up, please.
Yeah, we'll make sure everybody knows how to get in touch with you, Joe. Thank you for coming back. We really love everything that you've done. You've been so generous helping us understand these next steps, and we look forward to staying in touch and watching your continued growth and bringing along the rest of us as you continue pioneering in so many of these areas. Thank you.
Definitely. Thank you so much, Mary.
Thank you. Good to see you. Thanks. Okay, so you know where you can have your friends go and get jobs, but there are other companies here that are also hiring. So we just want to make sure you're all aware of that. What we want to do now is get going with what you've been waiting for, which are the presentations from the ventures. I want to introduce some of the two moderators that we have here today. We have both Deborah Novick, who is here from Westchester County, the Office of Economic Development. She's in charge of entrepreneurship and innovation. She's been awarded some New York State and probably soon regional ones for her economic development leadership. We're very pleased to have a chance to work with her. She has a background both in marketing and in finance. She's been involved in venture capital as well. So this is a really unusual background for a economic development and innovation background, but it's also kind of a sign of what we're seeing and hearing about from Joe about the maturing of the community. Nick Gunkel is here. He is a managing director at CTNext. He's in charge of the programs at CTNext. Both Deborah and Nicholas are responsible for the majority of the funding for the WCBA and ABCT programs. To the point that Joe was talking about with North Carolina being a place where there are a lot of workforce development programs, that is back in Nick's bailiwick, where he has been, prior to coming to CTNext, working on developing entrepreneurial talent within smaller cities, and in particular in an organization that's based in North Carolina and is tied to some of that development of the economy. So we're thrilled to have both of you. We are going to look forward to now having some pitches. Maggie's going to be bringing on the next people who will be starting to pitch, and we look forward to those presentations and your introductions. So I will turn it over to Maggie. We'll get some chairs off the stage, and we'll get going with the presentations. So thank you. Thanks, Rudy. Yeah. We have Ashkan?
Yes, I need your mic.
Okay.
Well, it's a pleasure being here. Thank you so much for coming. My name is Nick Gunkel. I'm a bit older than the picture suggests, but that's a beautiful picture, way back. It's my pleasure to introduce Ashkan Novin from Genesis. Ashkan and the team at Genesis are developing a CRISPR-based therapy to prevent cancer metastasis and recurrence. Join me in welcoming Ashkan.
Thank you. Hi, my name is Ashkan Novin, CEO and co-founder at Genesis. The story of our company is coming from a decade of research in cancer metastasis. In Genesis, we are developing a new level of CRISPR-based therapeutic to reduce the risk of cancer metastasis and recurrence. After treatments, there might be some cancer cells left behind in a patient's body. Those cells can grow again and form another tumor. If it takes longer, they will spread and form new tumors in patient's body. Current tests are not sensitive enough to detect those remaining cancerous cells, and the cancer that comes back can be harder to treat. The same therapy isn't always effective again, and the tumor can develop a tolerance to a certain treatment option. That's why the oncologists have to try other treatment approaches, and a patient has to do the follow-up treatments, which can cost around $35,000 only in the first year of the treatment. Let's talk about one of the most prevalent cancers with high recurrence rate, breast cancer. 1 in 8 women has a risk of getting breast cancer in her life. Every 13 minutes, a woman dies from breast cancer in the US. And more than 90% of those cases are because of cancer metastasis. Today, I'm introducing a new level of CRISPR-based therapy to make the local and healthy cells more resistant against cancer, to reduce the risk of cancer metastasis and recurrence, to reduce the time and cost in the treatment plan. With an intratumoral injections, we want to empower the surrounding tissue against cancer. But let's see how it works. We want to take a skin biopsy from a patient. After fibroblast isolation, we want to target a specific gene by using CRISPR and a single guide RNA. After expansion and quality control, we inject those cells at the tumor site. Now those cells are resistant enough to contain the cancerous tumor within its primary location, so the metastasis cannot happen. We have the cancer biologists working in this field for more than 15 years. This idea is led by Dr. Kishitis, a faculty at University of Connecticut Health Center. Dr. Solène is our bioinformatics expert, and we have the chance to have Dr. Gunther Wagner, a fellow of American Association for Advancement of Science on our advisory board, as well as Dr. Daneshmandi, Dr. Anna Louie, and Dr. Stevenson. This idea was started around 9 years ago. We have been doing our in vitro studies since 2018. All the preclinical studies have been done with the real patient samples that we received from UConn Health Center, Yale University, and Hartford Healthcare. Our animal protocol was approved in 2021. Now we are planning to submit our SBIR and STTR grants in the next cycle, and our patent by the end of this quarter. Our plan for the FDA regulation is already prepared and established. In our growth strategy and potential revenue in the future, we want to take a piece of that biopsy from a tumor. We sequence it to get the genetic information of that patient. And based on the results, and since we'll be running different programs in Genesis, we'll say which target and gene would be the most effective one for that specific patient. Now we are finalizing our second round of animal studies, and hopefully by the end of this year, we can get the efficacy data and we can optimize our drug delivery methods. And by the end of next year, we are planning to submit all the IND requirements and documents before going through the first phase of clinical trial. And to achieve these 2 milestones, we are raising $3.9 million. We want to make therapy more effective. In Genesis, we want to stop cancer and save lives. Thank you. I would be happy to take any questions.
I think we've got time for one question from the audience. Lucky one.
Thank you. Really interesting approach. I'm just worried if you're trapping the cancer in the tissue or the organ, is that going to be bad for the local cancer while you're protecting everything else?
That's a great question. So we are saying more than 70% of the cases for the cancer in the different cancer types, we can treat the cancer with knife, with surgery. As long as we trap the cancerous tumor within its primary location without any metastasis, it should be treated. And as I mentioned, more than 90% of the cancer deaths are because of cancer metastasis. So that's our main goal and highest priority, to stop cancer invasion and metastasis, even the micrometastasis at the tumor site.
Awesome. Thank you so much.
Thank you. Thank you.
It's my pleasure to introduce Udith Ashok from Piezo Biomembrane. Udith and his team develop and commercialize a new class of biodegradable and implantable material in the field of animal health. Welcome, Udith.
Imagine a life where every step you take, where the most mundane task, such as opening a jar, becomes a daunting challenge. For half of you in this room, this does not have to be an imagination 20 or 30 years down the line. Osteoarthritis affects 1 in 2 adults across the United States, and just in the United States, 32.5 million adults suffer from osteoarthritis by the end of their lifetime. And it gets worse. There is no current cure for osteoarthritis, and the treatment methods that do exist only address the symptoms and numb the pain. We are here to change that. Who are we? Let me introduce you to my team. Tan is a full-time professor at University of Connecticut. He is our co-founder. Previously, he worked as a postdoc at MIT in Bob Langer's lab. He's also a Princeton grad. Lin Lei is our co-founder. He is a Columbia grad, and he raised over $2 million for his last startup before making a successful exit. Me and Tin are recent UConn graduates. We graduated on a full merit scholarship, and we are really excited to be involved with Piezo Biomembrane. It has been known for a long time that electrical stimulation can regenerate hyaline cartilage. There are treatment methods out there which provide external stimulation to treat osteoarthritis and regenerate hyaline cartilage. We also are aware that piezoelectric materials, when squeezed, twisted, or otherwise deformed, can generate electricity. Our team at UConn developed a biodegradable membrane that has been FDA approved for implantation in humans and animals, which can generate electricity when implanted at the site of osteoarthritis and treat osteoarthritis by regenerating the hyaline cartilage. We have tested our product on animals. We have carried out extensive tests on rodents, and the results are promising. We obtained statistically significant results. We'll be passing along the research papers and also the presentation slides for further evaluation. The market opportunity is huge. We initially intend to enter the market by targeting the companion animal market and the equine horse markets, because regulatorily, they are less scrutinized as compared to human markets, but we do intend to expand into the human markets, which represents a market opportunity of around $80 billion. Our commercialization plan is set right now at this point to establish a UConn tip space. We are in the process of transferring the technology out of the academic lab onto the tip lab space, and we are also in the process of establishing relationships and performing customer pilot studies to market the product. So what are we looking for today? We are looking to raise around $1.2 million for tests in animals and larger mammals, such as horses and canines. And we're also looking for a community and people of like-minded individuals who have a vision for future of material in healthcare. Thank you so much. We'll be distributing our business cards, and we'd love to have a conversation. And me and Lynn would love to take on any questions that you may have.
Similar rules as last time. One question from the audience, but I can't resist saying I love the canine market. We have a Bernese Mountain Dog at home, and he's not there yet with the ailments, but hopefully you guys are going to be in time. Please.
You said the product or the membrane was FDA approved. What is the indication it's approved for?
It is approved for use in humans and animals, but it has not been approved for treatment in osteoarthritis. So that is the reason we are raising funds. We have clinical tests justifying that it could be used for treating osteoarthritis. And since it is biodegradable, it is approved for implantation in humans and animals, but it has not yet been approved as a treatment for osteoarthritis. But it can be implanted to generate electricity and treat osteoarthritis, as we demonstrated in our tests. Then, if you'd like to add anything.
Thank you.
Thank you.
The next company that we'll welcome is Life Farms. Ed and Life Farms are developing a novel antifungal compound, both active in vitro and in vivo, against drug-resistant fungal infections. Welcome, Ed.
Okay, thank you. Consider that you're a cancer survivor and have had a bone marrow transplant. And you're one of the 20% of bone marrow recipients that ended up in the hospital with severe fungal infection. And if you find out it's a drug-resistant fungal infection, your life is in danger. Life Farms has a compound that's for you, I think. I founded Life Farms in order to conduct a high-throughput screening, and we are developing a compound that specifically targets fungal resistance mechanisms. I started Life Farms. I used to work at Pfizer. Before that, I was at University of California at Irvine. And then I went to school at Washington University. I've worked with Timo Alaska, who's a chemist at Connecticut College, and Makbu Ganong, who's head of the Center for Medical Microbiology at Case Western Reserve University. The current problem that we have is fungal resistance is increasing. The number of people that are getting infected has increased because of the growth in the population of immune-suppressed people. There were 2 drugs before previous to last year. There were 2 drugs, azoles and echinocandins, were using that were treated these patients. However, resistance has grown tremendously in all in both of the drugs. Azoles used to be the primary treatment, but they are no longer used in many cases. The compound that we've discovered is codenamed 10581. It targets a mechanism that fungus use to maintain drug resistance. It's specific for that. It's a very sought after target, and it's talked about a lot, but it's not. We've currently, we are funded by an SBR1 and an SBR2. We've isolated the compound, and we've identified the structures. We've made several analogs. We've selected a candidate based on its in vitro activity, in vivo activity, and safety, and we're in the process of filing the patent. Going forward, we plan to recruit a CEO so that I can spend more time with the science. We maintain additional funding through various sources. We want to file an IND for the compound. We've got about 1/3 of it done, but I think, and meet with the FDA, but I think it's going to take about $2 million to get that far. We have clinical trials planned if we need to be. I just want to show a little bit of data on the compound. This is survival in mice, and these mice were treated with these drug-resistant strains. The first 3 panels are IP administration of the compound, and the 4th panel is oral. You can see the untreated animals, they received a massive dose of fungus. The untreated animals, the light green, they were just infected, and they were all dead after from 3 to 6 days. The other colored lines are increasing amounts of our compound, and you can see that in the highest dose, which is a very reasonable dose, there was no deaths at all. No deaths at all. The interesting thing is the animals received 3 doses an hour after the infection, and on day 2, day 3, and day 5, and we followed them for 8 days. There's a historical reason for doing the experiment that way, but it certainly indicates the compound it stays around for a long time because they were able, at 8 days, 3 days later, they were able to have it. There are no classes of drugs that have been approved, but there are 2 drugs that are approved recently in last year.
I think it's time to go.
Thank you.
Question from the audience? Otherwise, I have one. Just double checking. Okay. Oh, sir, you just rescued me. No, I got this question, so I'm not personally identifying too much with it yet.
Can you say what the target is that works against all these different mechanisms?
Yeah, I have a poster, but there's no place to put it up. The target, it targets fungal-specific heat shock 90 protein. There's a lot of the fungal resistance mechanisms are in the enhanced pumping out of the drug, or they have a resistance target in developing a resistance to the compounds. And most of them are maintained by heat shock 90. But mammalian heat shock 90 is I mean, there's a lot of trouble developing those compounds because of toxicity. But a fungal-specific heat shock 90, they have about a 300-fold, 200-fold selectivity for fungal over human. So we haven't had any toxicity results.
And are you as interested in, as I am, is what happens after 8 days?
Pardon me?
8 days seems awful close, awful short, Why'd you stop there?
Well, because that's the protocol. I mean, I didn't do these experiments that were done by Mahmoud Ganag in Cleveland. The experiment, what they did was they took the target organ, which was the kidney, and looked at colony-forming units per milligram protein. And 8 days was their standard at the time. I don't have enough money to do it any longer right now, to tell you the truth.
Sneaky how you added a second question to this. I love that. There's a minute left. Like, I'm too fast. I'm trying to make up time. Beautiful. Thank you, Ed.
Okay.
I think it's you.
Yeah. Well, now it's my pleasure to welcome Rashid Rumah from Nanolytics. Rashid and Edmund are dedicated to stopping the devastating consequences of multiple sclerosis at its initial onset. Thank you.
So we are Nanolytics, and we are interested in treating multiple sclerosis by targeting its root cause. So we hypothesize that MS may be caused by gut-derived neurotoxin secreted by Clostridium perfringens. And we have engineered state-of-the-art camelid nanobodies to neutralize this toxin with potential entry into humans by Q4 of 2025. Our work is protected globally by a granted patent, and we are seeking $4.75 million of tranche investment. And importantly, there are no current direct competitors targeting epsilon toxin in the context of MS. So MS affects the blood-brain barrier and CNS myelin, and the cause remains unknown, making it very difficult to treat, and there's no cure. In 2013, we published our first paper showing that ETX, epsilon toxin, binds to blood-brain barrier vasculature and to CNS myelin. So that's depicted here, blood-brain barrier vessel stained in green and epsilon toxin fluorescently tagged in red. And if you look at this magnified vessel, you see the toxin lining the vessel, the lumen of this vessel. Once it breaks down the blood-brain barrier, it now has access to the brain, and now it binds to myelin. So here we have a mouse corpus callosum with myelin stained in green, and fluorescently tagged epsilon toxin stains the same structure. So in 2013, we also had the first case of an MS patient carrying the bacterium in their fecal microbiota. And fast forward 10 years later, we currently have the cover of JCI, the current issue of JCI, and this new study shows that we can now detect this in 61% of MS patients compared to 13% of healthy individuals. And if you compare an MS patient carrying this bacterium to an asymptomatic healthy carrier, the MS patient carries at above 1,000-fold. So there are multiple key opinion leaders supporting this work. Most notably would probably be Alfred Sandrock, who is the former head of R&D at Biogen IDEC. And just to summarize the hypothesis, this gut bacterium is really not typically infecting humans. takes up residence in the GI tract, secretes epsilon toxin that's absorbed into the circulation, and then it binds to the blood-brain barrier, breaks it down, then it enters the brain and binds to CNS myelin. And it also allows inflammatory cells to enter the brain and exacerbate that tissue damage. So we've engineered camelid nanobodies to, one, block the toxin at the site of origin, but also we can recombinantly engineer these nanobodies, fuse them to human FC, and make an antibody-like molecule to block the toxin in the circulation to protect the brain. We can also fuse these nanobodies to affinity tags to capture epsilon toxin in MS biospecimens, and we think this study will be critical to validating the hypothesis to show that the toxin is present in their biospecimens. So here's our milestone sequence. We have identified 3 nanobody sequences, 2 of which are neutralizing, 1 of which is high affinity, but non-neutralizing that we will use for diagnostic processes. And we're interested in testing the efficacy of these neutralizing nanobodies in animal studies, optimizing the administration route and dosing. We're going to go with a single lead candidate, and then to move forward for a pre-IND meeting with the FDA. We're asking again for $4.75 million to move through that process. So we are protected in all the major markets globally. And just to introduce you to our team, spearheaded by Dr. Edmundo Campisi at Rockefeller University and myself, Rashid Rumah, also at Rockefeller University. So I'll take any questions at this point.
Go ahead.
It's a very intriguing concept. I'm just curious, why nanobodies versus, say, a regular antibody?
Yeah. So we do have neutralizing monoclonal antibodies as well, but camelid nanobodies have the added advantage of being able to recombinantly engineer them any way that we want, and also to be produced in bacterial cells as opposed to mammalian cells for humanized monoclonals, which will really reduce the cost and basically allow us to, I think the main advantage is really the ability to fuse it to whatever you want. We can make multivalent nanobodies to increase the avidity of that molecule to make a better binder. But I think also it gives us the flexibility to treat, let's say in the gut, right, with those nanobody fragments where you don't want the human FC, but you also have the option of fusing it to human FC and treating in the circulation like a more traditional humanized monoclonal.
Would this be more of a preventive measure, or is that a treatment against MS?
Right, so the way that this would probably work out in the clinic is, you know, a patient presents with their first neurologic symptoms, maybe it's double vision, they get diagnosed after an MRI, and then you put them on an infusion with a blocking antibody or nanobody. It wouldn't make sense to deploy these without any neurological symptoms as a prophylactic.
So is that bacteria constantly present in the gut, and they have to constantly be targeted with a drug?
Yeah, so we're actually working on, the answer is yes, and we are working on novel strategies to try to decolonize the MS GI tract of the bacterium. But for right now, it's really just blocking the toxin, since we have good reason to think that is there throughout the extent of the disease.
You don't happen to have found any data where multiple sclerosis patients were treated with broad-spectrum antibiotics and their symptoms improved?
So the literature is a bit mixed on that. This bacterium is sensitive to penicillin. And with the penicillin literature, there's some studies that say that it was protective, some studies that say it was deleterious. So I think when you're working with the gut microbiome, which is a very complex environment, you can wipe out species that are natural competitors and make it more favorable for the bacterium. And also, it's a sporulating bacterium, meaning that those can survive antibiotics, basically. So it's complicated.
Thank you so much, Rashid.
Thank you.
Our next presenter will be Cathy Beaumard from Nervosave Therapeutics. Nervosave is based in France and New York. and is focusing on the prevention and cure of myelin-related disorders. Thank you, Cathy and Nicholas.
Thank you. Hi, everybody. I'm very proud to introduce you to Nervosafe Therapeutics with Nicolas Tricot, the CEO and CSO and founder. Nervosafe Therapeutics is the world leader in prevention and in preventive and curative gene therapy for rare and common peripheral nerve diseases. We want to build a business in the US with a CEO in the US. Our lead program, NVO-101, is designed to treat CMT1A. Highly disabling, as you can see, without any treatment on the roots at the moment. With more than 68,000 patients in the world and in the US, and EU5, the disease is very disabling. CMT1A is due to the duplication of the PMP22 gene. Our solution, NVO-101, is an AAV9 and RNAi-based gene therapy that reduces the abnormally high level of the PMP22 protein. We propose a single injection in the main nerves of legs and arms. It doesn't modify the genome. Nervosafe Therapeutics has already validated NVO-101 in the mouse model by decreasing the PMP22 protein by 40 to 60%, and in the monkey by 25 to 40%, high enough for efficacy and to maintain the physiological conditions. NVO-101 is fully preventing the disease in the mouse and rat models after a single injection. NVO-101 is a safe product, and local injections are very well tolerated by monkey. Unique and the most advanced gene therapy in development, NVO-101 is the best-in-class disease-modifying solution for CMT1A. And furthermore, for our lead program, we have a strong IP protection. Our 35 million Series A will allow us to bring NVO-101 through phase 1-2 clinical readouts, and the inflection point is the first clinical validation in the second quarter of 2025. Nervosafe Therapeutics, it's a management team with Nicolas Tricot, with me, CEO, with Sarah Abbas, our Chief Medical Officer, and Julie Espalerg, our Discovery Research Director. And also completed by experts, international network with a scientific and clinical board, a strategy board, and supports American and French in business, IP, and also manufacturing. Nervosafe Therapeutics is raising $35 million Series A to develop a cutting-edge technology to treat CMT1A, and with an exit for the investors within 4 years. It's the first-in-class myelin-focused international company with a robust IP and its lead program ready for phase 1-2 next year, in fact. NVO-101 peak sales of $1.7 billion should be by 2033. Thanks for your attention, and we are waiting for your questions. And sorry for my stress.
Do we have any questions in the audience? If you do, wave your hand vigorously. It's a little hard to see with the light at my eyes. Do you see someone?
Way on the right.
Yes, go ahead.
So it sounds like you're proposing a one-shot cure, is that correct?
Yes, it's correct. It's a single set of injections.
Yeah.
In all the main nerve of legs and arms, to bring benefit to all muscle of legs and arms.
Any other questions?
Okay.
I do have one question here.
Yes, sure.
What applications are there for your gene therapy platform technology beyond NV101 and CMT1A?
Yeah, so we are actually developing a new gene therapy platform in order to prevent demyelination of Schwann cell in peripheral nerve. So that should be a gene therapy that can be applied to all CMT, demyelinating CMT disease, even the ultra-rare one. So you know you have sometimes 10% in the world that have a problem. You don't know what is the problem. You know it's demyelinate. You can use our gene therapy to prevent demyelination in this kind of disease too.
Thank you very much.
Thank you. Thank you for your time.
Thank you.
Oh, this is out of order. Chris, are you presenting now? So I happen to know that this is Chris Buser, and he'll be presenting now on BioCubic. And I don't have the little blurb, so I can't say exactly what you're doing, but you'll tell us.
BioCubic delivers high-resolution nanoscale tissue data to improve the precision of research, drug development, and the diagnosis of diseases. So why do we need nanoscale imaging? Think of kidney disease diagnostics. So magnify 1 million times, and now your kidney is the size of Long Island. And your job is to find the houses that are structurally deficient. So you drive around, you check out the neighborhoods, you walk up to a house, you check the exterior, but what you really want to do is you want to go in and inspect the foundation. We do very much the same thing in diagnostic tissue imaging, using histology to get to the microscale and check where the cells are in all of this. The problem is that we stop at this point because the next step to the nanoscale is done by electron microscopy, which is extremely tedious and very expensive to do. And we stop there even though we know that this nanoscale data dramatically contributes to the precision of the diagnosis. And unfortunately, that doesn't only happen in diagnostics, it also happens in basic research and in drug development. So I founded BioCubic to develop nanohistology, which is an add-on technology to the traditional histology that gets us to the nanoscale. It's 1,000 times better than histology, and it's at least 10 times simpler than electron microscopy. And this combination allows us to make this nanoscale data available to millions of people instead of thousands of people. Our strategic plan is to begin with the MVP, which we can sell into the R&D market. We then add higher efficiency and higher volume features that get us into clinical trials and the diagnostics market with a data component later on. BioCubic has the only solution that delivers nanoscale data at a scale large enough to train AI on this data. And there is a lot of activity in that in the histology space, which is great because we're an add-on technology and we can adopt some of their solutions for our purposes.
Yeah, point that way.
There you go. We have a fantastic team. Myself, I have 25 years of experience in electron microscopy services, and my partners complement me beautifully with their expertise in business. We plan to reach break-even by the end of the first year through the sale of services and keep growing by adding the higher volume markets. The plan to develop the MVP is through a blend of revenue grants and the seed investment, and that will get us to the MVP with sales. We are already We already have a microscope in place, and we're already marketing our starting imaging services, and we've already applied for non-dilutive funding. So I hope I could convince you that nanoscale data is really the foundation for precise decisions in biology, and I'm happy to take any questions you may have. Thank you.
Do we have any questions for Chris about BioCubic? Arthur, I was counting on you because he said kidney disease. Go ahead, Joe.
Just for my own understanding, so with the nanoscale, how do you sample enough sample size to get a representative picture of the tissue?
That is a very good question, and that's actually one of the problems. So when you go back to the Long Island picture, right, this becomes a needle in a haystack problem. It's very, very challenging because of the scale of the problem. So it's really about acquiring a lot of data in a lot of different points. And once our technology is automated, we can do that fairly easily. And you can combine that with machine learning, right, where you tell the algorithm, go and look for these features in different areas. But it definitely requires a lot of imaging time.
Any other questions? Top left. Okay, top left, I see one hand. Go ahead. Yes, go ahead.
Hi, I'm interested, are you using the materials to label, the label and materials to sequence? Is it nanomaterials or something different?
So that is the beauty of this technology. We use atomic force microscope. Think a phonograph needle that scans the surface of the sample. So it's completely stain-free. There is no staining. You image the biology directly with the interaction with that needle.
And you're able to see molecular level?
Yes. AFMs go down. You can image DNA with these systems. You can see individual proteins. So it's not a limitation of the instruments. It's a limitation of the preparation protocol. But yes, you can.
And you said it's atomic force microscopy.
Atomic force microscopy, correct.
We have no more time. Okay, thank you so much, Chris.
Thank you.
It's my pleasure to introduce Ali Kabiri from CellSpin. Powered by AI and novel photonics, Ali and the team at CellSpin reliably detects a single tumor cell among billions of healthy ones.
Thank you. Good afternoon, everyone. CellSpin is building the next generation liquid biopsy technology. This is Ali, CEO and founder of CellSpin. CellSpin solves three major problems of existing liquid biopsy solutions: lack of accuracy, long wait time, and high cost. Unlike most existing liquid biopsy solutions, we're not looking for remnants of dead cancer cells. Rather, CellSpin analyzes billions of fast-flowing cells to detect extremely rare cancer cells at very high accuracy. To make this possible, our patent-pending breakthrough photonic solutions captures hundreds of images of every single cell. Then our novel generative AI combine all of these images and reconstruct a coherent atlas or map of protein expression on the cell surface. This comprehensive view allows us for the first time to detect cancers very confidently with very minimal false positive, even if not any. This is how our end products looks like in practice. This state-of-the-art solutions can disrupt the market as the most sensitive, specific, scalable, and accessible for liquid biopsy solutions. So CellSpin has the right team of experts to deliver its mission. Nima and I were 27 years friends, and then we founded this company. I have 9 years of experience co-inventing the first digital protein sequencer on a chip and leading the photonics division from company inception to its IPO in 2021. Nima was AI lead at Apple, and then he previously founded 2 other startups in AI and machine learning. Amy is a cell biologist from Cambridge with 20 years of experience in startups, and Peter is one of the founding members of NVIDIA. Our team has a seamless support of world-class leading experts in business, medicine, and technology from MD Anderson, Caltech, and Stanford. Our working proof of concept is capable of detecting cancer cells with the accuracy of 1 in 40,000. And this is just in the start. Right now, we're working on our alpha prototype in a small form factor to put it in hands of experts in the leading cancer centers. And however, this is not our ultimate goal. Our ultimate goal is that to have our product widely available and accessible as a point-of-care diagnostics. So we will sell instruments, consumables, and AI platform subscriptions. In addition, we provide lab services to cancer centers and physicians. We have carefully designed our 5-year roadmap. Bottom line is that by 2027, we will be in production, and we will have our 510K clearance in hand. We're looking for $700 in investment on our pre-seed safe with a $10 million cap. Our founders and institutional VCs have already invested $400,000 on the same term. Thank you for your attention. Delightfully looking forward to connecting with you. Together, we can make a difference in cancer diagnostics and save lives. Thank you very much.
Very impressive team.
Thank you, Ali. Any questions from the audience?
I have a question, if no one has.
Yeah.
So I have a question. An important thing with any startup is how it fits into the workflow of the physicians or the hospital or the cancer center. So will this have to displace something they're doing? Will it have to fit into a small exam room? How will it make its way into clinical practice?
That's a very, very good question, and absolutely is a hard path. However, at this moment, we're in discussion with one of the leading hospitals for breast cancer. Our first, or I would say our starting practice would be on the breast cancer patient samples. The money that we're raising is to just get more samples and then make connection with that hospital. So we're talking with the head of the clinical trials, and then they're very happy, and the result that we provided to them, very liked it, and then said, we would love to have this one here and start collaborating with you.
So they'll show you how it can fit into their workflow, and you'll adjust around that.
Totally. And at the same time, we have a support of MD Anderson, so the leading team that we're working with them, very supportive. And then for the CLIA lab, we definitely want to work with them and then have in their CLIA partnership.
Good luck to you. Very fascinating.
One more question we have. Sorry, I'm going to steal that one. Can you perhaps elaborate a bit why you believe the photonic system will yield a better accuracy in the diagnostic than some of the comparable or rivaling ways?
A very, very good question. So a couple of things comes from photonics. First is the scale in imaging the cells. That's for the first time we can image the cells hundreds of times while in there, and there's a fluing. So without that, it's impossible. You can take an image once, but we can do hundreds of images. So adding those hundreds of images and combining through the generative AI algorithm that we have, this is for the first time we can show the atlas or map of the protein expression on the cells. That is the key that it helps us to mostly reject all the false positive and then detect the actual cells.
Thank you.
Fascinating.
Appreciate it.
Thank you.
Next in line is Alexey Melnik from Aroma Science. Alexey and Aroma Science are developing the direct-to-consumer test to improve lives affected by metabolic imbalance diseases.
Hello, everyone. Did you know that we carry 3 kilograms of microbes in our gut every single day, all the time? Collectively, we call those microbes our gut microbiome. They have 100 times more genes than we do, which allows them to perform many useful functions, including producing vitamins, helping us digest food, and even produce neurotransmitters to regulate our mood. When everything works well, they work in a perfect harmony with our bodies. But when things go out of whack, then the gut enters the state of microbiome imbalance. This can be caused by many factors, including antibiotics, lifestyle, and exposure. When this happens, this causes metabolic imbalance and low-grade inflammation, which in turn exacerbates microbiome imbalance. This vicious cycle has been linked to many chronic diseases, including cancer, type 2 diabetes, and cardiovascular disease, and many others. We now know that not only the microbes that are important, but also what chemicals they produce are the most important thing to measure. My name is Alexey Melnik, and I'm CEO and co-founder of Aroma Science. 11 years ago, I joined the lab of Peter Dorstein's lab at UCSD. Then Alexander joined, and we formed the company. We've been publishing a lot of articles in many reputable journals with a lot of citations on the topic, and we developed many tools to understand microbial chemistry. Established 5 years ago, Aroma has been providing those tools to our biotech and pharma companies, customers. In particular, we provided actionable insights into biological systems, such as microbiome systems, to understand drug and probiotic metabolism, but also patient stratification. But what's most important, we provided biomarker discovery platform. You can think of a biomarker discovery if you take a complex biological sample and boil it down to biomarkers of diseases. This is what we've done. We discovered novel microbial metabolites recently that were implied in IBD and NFLD. We even patented those molecules as potential therapeutic leads. And then we understood, when we realized that this type of analysis is very important, but it's rarely accessible direct to consumer. And this is where we decided to develop direct-to-consumer tests, which will rely upon 3 major pillars of testing, such as urine, blood, and stool. For urine, we have a proprietary dried blood urine card, which person can collect at home and send us over a regular mail without cold storage for us to understand concentrated biomarkers. For blood and stool, we're partnered with the top-tier producer of supplements in the US to screen for systemic health in blood using the OneDraw device, which is a painless and non-invasive, and stool swipe, essentially to understand microbial metabolism. We believe these 3 pillars of testing will allow us to get insights into one's health and create an AI-based predictive solutions for lifestyle and dietary changes to prevent, reverse, and even cure chronic diseases. Today, we're generating revenue from our B2B platform. By October, we're planning to validate all these tests. In April, we're planning to collect large budgets of data for healthy volunteers as well as disease cohorts. And then by July 25th, we're planning to launch the test in the app to consumers. We'll be proud to support it by a strong operations team in place and also a strong board of advisors who are experts in AI and chemistry fields. But we're planning to expand this team to be able for us to help us to achieve our direct-to-consumer test goals. With that, I would like to thank everyone for the attention. I'll be happy to take questions.
Any questions from the audience? Please, sir.
Hi, Alexey. Can you hear me?
Yes.
Okay. Thanks for a great presentation. Can you tell us a bit more about the target molecules that you've discovered and what their relevance might be in disease and health?
Hey, incredible question. So we discovered novel bile acids. So we now know that there are at least 200,000 microbial bile acids that are in the gut that are produced by bacteria. Primarily, we discovered those that are produced by Clostridia and that were implicated in FXR agonism pretty well. Those that we discovered were 20 times more agonistic and more effective in binding to FXR than the most potent known to humanity, which is chenodeoxycholic acid. So those we found. And we have a specific panel that we're targeting here of at least 25,000 right now, so bile acids. Please.
Do you feel like it might be hard for the consumers to interpret the complexity of the results?
Indeed, yeah, this is what we're going to be working mostly on, so that to deliver the data in a way so that people understand what to do. We think that the most important thing is not to show the reference range of some people that might be not really important in the context of one's health. We think that what's most important is democratize the test, make it so cheap so that you can sample yourself throughout the lifespan, so that you can create the reference range for yourself when you're healthy, ideally, and then you can monitor yourself for over time and see what biomarkers are changing. And then the AI predictive tool will allow us to predict what kind of interventions were done and what needs to be done to be able to go back to the healthy state.
Yeah. Got time for one quick one? Yep, gentleman in the back.
It's not just for dysbiosis, but this is a start. So for dysbiosis is the way to start. It's actually really hard to detect dysbiosis until you know what biosis is, because we're all different in types in terms of what microbes we have in the gut. So it's really what we want to see is, what we want to do is a longitudinal sampling, and then we have a set of predictive biomarkers for a wellness score and test, and then if you deviate from those, then we'll tell you what is that you've done, or what is that you need to do in order to bring it back. But it's not only for dysbiosis per se.
Is there time for one more? So can I ask a follow-up to that? So if you need longitudinal data to understand what someone's own biosis is, wouldn't it make sense to involve the doctor so that, for example, you could have these tests as a routine matter, even from your pediatrician, from your internist, so the data would be developing for yourself from a healthy state over time, naturally, without waiting for me to go to have the idea of doing this?
Oh, absolutely. So this is, as a matter of fact, this is how we're going to start. It's going to be through point of care, physicians, direct to consumer, and then people can also screen themselves later down the line, but that's the start.
How often would somebody need to be screened to
I think the ideal, I don't have exact answer to that now. My feeling is that it can be once a month or so. And we will make the test so that it's cheap enough so that you can do once a month testing.
Really interesting. Thank you.
Thanks so much.
Thank you.
Last before intermission is Dr. Solène Constantini with Hack Your Care. Solène is technology that optimizes healthcare professionals and a corporate need for medical expertise in health R&D.
Thank you. Hi, everyone. I'm Dr. Solène Constantini. I'm a surgeon from France and the CEO and founder of Hack Your Care. Imagine that today you are a company and you want to get to access fast medical expertise with clinicians from the field. How do you do? Now, imagine you are a physician, you are working in a hospital, for example, and you want to access to be part of innovative projects. How do you do? I have designed this solution, Hack Your Care, which is a unique platform powered by an intelligence engine to optimize the connection between healthcare professionals and the corporate need for medical expertise. So we are building this two entries platform, one for physicians, so they are profiled by skills and commitments, and the other entry, which is for companies, and they can put and log in, make a deposit of their project, and this project is going to be qualified to go to the next step of the product development. So for example, a medical device company, which needs testing, user test, they can log in, put the project, and with a lot of items, we can qualify for them the perfect step they are, and qualify for them the medical experts they need. So we are working today on a business model with two revenue sources, one which is already active. We are doing like a marketplace, so a commission-based revenue stream for placement of clinicians from our pool of talents. We have already 2,000 clinicians from France, Europe, and US. A second one, which is coming soon with our launch of our new product, a subscription business model. So we are working today with health tech companies. SaaS company wanted to address the health tech sector, but also deep tech company, and we are able to qualify them from the design phase, but helping them to go further by, with the help of the medical experts, to validation, clinical trial test, but also commercialization with ambassadors. So we have actually no competition, direct competition. We have also a research platform in the US, which are indirect competitor. So we have a big traction this month. We have already 15 clients in France and in the US now, and we have a well testimonies. So our first target market are French, Europe, and also US next year. Our roadmap is that we have already made a first fundraising last year with business angels for almost all our clinicians in France, and we are going to launch our new product after 10 months of development in 2 weeks. So this is our team, combination of multiple skills with a tech, so expert tech advisor, Nicolas, and a growth advisor marketer, Xenia. We are looking for 1.5 million to be able to build this growing team. We have already, but we want to be able to pay them growth, and so we will need also this money to our product development. Thank you very much.
Question for Solène. I've got a question. Oh, yes, thank you.
So is it more of a subscription-based model, like you would be able to sign up and access this as often as you need to, or is it on a service kind of sales?
So this subscription business model is based on our product we are actually building. So we want companies, big companies like we were exploring with big companies which already are working with a lot of clinicians, and they would like to use our AI within their proprietary communities. So it's more bigger companies, and that's why we have this both possibility of business model today. We have also already 3 clients which are waiting for this new business model. Yeah.
So I can see the value of being able to find the right clinician, so that's fine. I also wonder if there's value to your numbers, as in if I want to be able to reach, for whatever reason, 100 clinicians interested in my area, and you have them in your database. Can you leverage your database in other ways?
Yeah, so we are not only a database, because we are interesting healthcare professionals to come on our platform. So that's really the difference compared to our competitors, because we are interesting them, clinicians from the field, to participate to this project, so they are clearly more committed, they are really happy, and they are giving like that more information about the product.
Awesome. Thank you so much, Solène.
Thank you very much.
Well, what a wonderful way to start off. We are going to take a 20-minute break right now, and then we'll return with some more innovations and breakthroughs. So please join us. The ventures who have just pitched all have easels, so you can look at the names of the ventures, and you can find them. They'll be at the top of the stairs here in the area right at the bottom of the escalator when you came in, and we'll be back in 20 minutes. So thank you very much, and we'll see you outside. Thanks.